Novavax COVID-19 Vaccine vs Merck COVID-19 Vaccine. This video is part 5 and 6 of 7-part series of Road to Vaccine Manufacturers, brought to you by HealthyRN Ways. Videos will be posted every Monday, Wednesday and Friday. Please like, share and subscribe to my channel to see the part 1, 2, 3, 4 series. See the link below for more information. Novovax COVID-19 vaccine is the 5th manufacturers who are doing a clinical trials for the COVID-19 vaccines. Merck COVID-19 vaccine is the 6th manufacturer who are also doing the clinical trials. Merck stopped its COVID-19 vaccine development, so its vaccine candidates will not be made available. Here is a peek of Merck COVID-19 vaccine. Novovax is a Maryland-based biotechnology firm that is taking a traditional approach to developing a vaccine against the coronavirus disease 2019, COVID-19. The company began a phase 3 trial of its vaccine candidate, NVX-CoV-2373, in the United Kingdom in September and a second phase 3 trial in the United States and Mexico in December. These two late-stage trials will enroll a combined total of about 45,000 participants. While this puts Novavax behind its competitors, initial results of its vaccine trials shows promising. How it works: the Novavax vaccine uses a custom-made spike protein that mimics the natural spike protein in the novel coronavirus. Novavax has for years worked on developing its recombinant nanoparticle technology, and created the first COVID-19 vaccine using this method in the spring. By summer, early clinical trials showed that the vaccine appeared to be safe, and more advanced trials entered the planning stage in the United States and other countries. The two-dose vaccine works by introducing a protein that prompts an antibody response, which blocks the ability of future coronavirus to bind to human cells and prevents infection. The protein is combined with Novavax's matrix M adjuvant, which is important in enhancing the immune response elicited by the protein antigen. How effective is it? Early vaccine trial results published by Novavax revealed a strong antibody response to the vaccine in animals. By August 2020, early human trials showed strong antibody responses with no serious adverse events and few side effects. In late January 2021, Novavax announced that a preliminary analysis of data from its UK Phase 3 trial showed that the vaccine was 89.3% effective and included protection from the UK variant B.1.1.7. The company also announced that the vaccine was 60% effective in a Phase 2b clinical trial in South Africa and included protection from the South African variant B.1.351. Results from all late-stage clinical trials are expected in 2021. In Novavax's trial, participants will not be directly exposed to the virus, but instead researchers will use a natural infection approach. The challenge will then be determining whether the vaccine or other protective measures like masking or social distancing offered the most protection. When will it be available? Novavax's vaccine candidate is generating a lot of interest because early animal study data showed that it was highly effective at preventing replication of the coronavirus in nasal passages. It is an important development for COVID vaccines since it may help reduce both infection and transmission rates. Although, this has not yet been established in human studies. Novavax is behind its competitors when it comes to clinical trials, so even if it continues to do well in trials, the vaccine may not be available until late 2021. The company has not released any estimates on distribution dates yet. The company itself also faces a significant challenge since 2019 was a difficult year for Novavax. Science magazine reported that the company sold some of its manufacturing facilities and has to rely on more contractors to manufacture many of its vaccines. This could result in a delay in production. No information has been provided yet about the price of the vaccine. Who can get the Novavax vaccine? Like other COVID-19 vaccine candidates, the Novavax vaccine has been tested in adults aged 18 and up. The company has not provided updates on trials in children and teens. Information hasn't been provided yet on concerns for specific groups. Regardless of study group populations, who can get COVID vaccines will largely be a regulatory decision made by the Food and Drug Administration, FDA. Eligibility during distribution will then be determined at the state level and by the Centers for Disease Control and Prevention, CDC. The CDC is leading vaccination efforts, and all orders of the COVID-19 vaccine, regardless of the manufacturer, will go through the agency. The CDC is overseeing the distribution of vaccines too. CDC's Advisory Committee on Immunization Practices, ACIP, has made recommendations on how to prioritize vaccine supplies. Although little information is available on the specifics of when everyone will receive the vaccines and where they can get it, state and local health departments will be coordinating efforts to distribute doses of the vaccines as they become available. The vaccine should be available both in physician offices and retail locations like pharmacies that administer other vaccines. Once the vaccine is available, any doses purchased by the US government will be free to its citizens. While the vaccine itself is free, the facility or agency that offers the vaccine may charge a fee for administration. Public health programs and insurance plans are expected to reimburse patients for any costs associated with COVID-19 vaccination, but no information has been made available yet. Side effects and adverse events. No serious adverse events were reported in early trials of the Novovax vaccine, but more information will be available after the late-stage trial results are published. In Phase 3, adverse events occurred at low levels and were balanced between vaccine and placebo groups. Funding and development. Novavax has been awarded more than $2 billion globally for its efforts to develop a COVID-19 vaccine, with $1.6 billion from the US government alone. The US funds were given as a part of Operation Warp Speed and from the Biomedical Advanced Research and Development Authority, BARDA. BARDA is a program that falls under the US Department of Health and Human Services. That program and Operation Warp Speed have sought to speed up how quickly vaccines to fight COVID-19 can be developed and approved for use. Novavax says its COVID-19 vaccine is 90% effective. Pharmaceutical company Novovax just completed its Phase 3 clinical trial in the UK. The company says its vaccine is nearly 90% effective at preventing COVID-19. The vaccine was much less effective against the South African strain of the virus. Pharmaceutical company Novavax recently shared results of its Phase 3 clinical trial in the UK. Its vaccine is nearly 90% effective at preventing COVID-19. Novavax revealed in a press release last week that its vaccine prevented 9 out of 10 cases of COVID-19 in its clinical trial in the UK. The vaccine was also effective against B1.1.7, the highly infectious strain of SARS-CoV-2, the virus that causes COVID-19, that has been heavily circulating in the UK. However, the Novavax vaccine had much lower effectiveness against the South African strain, B1.351. In a study of 4,400 participants in South Africa, the vaccine was only 49% effective. When the data only accounted for participants who did not have HIV, the efficacy increased to 60%. The Novavax vaccine, aka NVX-CoV-2373, is the first vaccine to demonstrate not only high clinical efficacy against COVID-19 but also significant clinical efficacy against both the rapidly emerging UK and South Africa variants. NVX-CoV-2373 has the potential to play an important role in solving this global public health crisis. We look forward to continuing to work with our partners, collaborators, investigators and regulators around the world to make the vaccine available as quickly as possible. The Novavax vaccine is currently in Phase 3 clinical trials in the US. What this means for you: the Novavax COVID-19 vaccine has a high efficacy rate overall. It's currently in Phase 3 clinical trials in the US and is currently not authorized for use in America. If it is approved, it could be another vaccine available to help reduce the spread of COVID-19. How the Novavax vaccine works: the Novavax vaccine works differently than the two vaccines currently approved for use in the US. Those vaccines, made by Pfizer BioNTech and Moderna, use a newer form of technology known as messenger RNA, mRNA. Those vaccines encode a part of the spike protein that's found on the surface of SARS-CoV-2. The mRNA vaccines use pieces of the encoded protein to create an immune response from your body. As a result, you develop antibodies to the virus. The Novovax vaccine uses an insect cell line. The cells were infected with a different kind of virus, called a baculovirus, and created spike proteins similar to the spikes on the surface of SARS-CoV-2. The vaccine contains several spike nanoparticles, which attract immune cells. Those immune cells create antibodies to the spike protein that can help prevent future COVID-19 infections. The way the Novavax vaccine works is not that different from our influenza vaccine. The parts are just slightly different. The Novavax vaccine is stable at normal refrigerator temperatures, making it easier to store and administer than the currently approved vaccines, a big plus for distribution. Novavax vaccine safety information. Novavax hasn't yet released safety information on its vaccine or given details about side effects. The company said in its press release that the vaccine cannot cause COVID-19 and cannot replicate inside the body. Novavax has not given details yet about potential side effects, but said that its safety database showed that severe, serious, and medically attended adverse events occurred at low levels and were balanced between vaccine and placebo groups. More research needed. While preliminary data sounds promising, stresses that there's not a lot of information available about the vaccine at this point. They have not even published preprint data. The scrutiny that the data needs to undergo hasn't been done by me or the scientific community. The lower efficacy against the South African strain of the virus is concerning, adding, "We need more details about that, including details about whether the vaccine performs better in younger or older people. The vaccine's efficacy against the South African strain of the virus, but is eager to learn more about the vaccine. But, overall, the vaccine is still highly effective at preventing severe disease with COVID-19, that's what matters. The end goal," he says, "is to turn COVID-19 into a cold. If we can get several effective vaccines, we can hopefully get there." An overview of the Merck COVID-19 vaccine. January 25, 2021, Merck announced that it will stop developing its two COVID-19 vaccine candidates, V591 and V590. The company stated in a press release that the immune responses were inferior to those seen following natural infection and those reported for other SARS-CoV-2, COVID-19 vaccines. Merck, one of the largest pharmaceutical companies in the world, focused its early efforts in the fight against the coronavirus disease 2019, COVID-19, on looking for ways to treat people who were sick. The US company also worked on two vaccine candidates for COVID-19 but announced on January 25 that it will stop developing them due to a lack of immune responses in trials. Merck continues to focus on its drug candidates for treating COVID-19, including an antiviral medication. The discontinued vaccine candidates are viral vector vaccines that use modified versions of other viruses to deliver instructions to cells. The company began enrolling participants in an early-stage clinical trial for COVID-19 vaccine candidate V591 in late August and an early-stage clinical trial for candidate V590 in late October. Merck was expected to share some preliminary results from the vaccine trials in early 2021 but instead announced the end of their development. Merck plans to still submit its studies of these candidates for publication in a peer-reviewed journal. How it works: Merck's vaccine candidate V591 is a recombinant vaccine that uses a live virus, measles, vaccine as the delivery method for transporting a small piece of the coronavirus genetic code to create an immune response. Recombinant viruses are genetically engineered viruses, which can be designed to act as vaccines when antigen genes are added. V591 was launched by Merck with the purchase of Themis, a private company focused on immunity and vaccines that Merck acquired in May 2020. Merck collaborated with the International AIDS Vaccine Initiative, IAVI, on the development of its other vaccine candidate, V590. V590 uses recombinant vesicular stomatitis virus, RVSV, technology, the same that was used by Merck to develop a vaccine to fight the Ebola virus. For this particular vaccine, Merck was using a common animal virus that has been changed to use certain proteins on the virus that help launch an immune response to the novel coronavirus. How effective is it? There is no data released yet on how the Merck vaccines performed, but the company announced in a press release that both V590 and V591 were generally well tolerated, but the immune responses were inferior to those seen following natural infection and those reported for other SARS-CoV-2, COVID-19 vaccines. Who can get the Merck vaccine? Merck enrolled 260 participants 18 and older, grouping them by those 18 to 55 and those older than 55, for the V591 trial. Merck's enrollment for the V590 trial was also 18 and older with similar age groupings. Merck is no longer developing vaccine candidates. If vaccine candidates from other companies are authorized, the Centers for Disease Control and Prevention, CDC, is likely to dictate who should receive the vaccine and when. Although little information is available on the specifics of when everyone will receive the vaccines against COVID-19 and where they can get it. Funding and development. Merck has received more than $38 million in funding from the U.S. government for its COVID-19 vaccine program as part of Operation Warp Speed, a public-private partnership initiated by the U.S. government to facilitate and accelerate the development of a COVID-19 vaccine, and from the Biomedical Advanced Research and Development Authority, BARDA. Merck stopped its COVID-19 vaccine development, so its vaccine candidates will not be made available. Even Merck vaccine will not be available. We still have 5 more manufacturers running the race for the road to COVID-19 vaccine. Which vaccine are taking or have taken? Whatever vaccine is available in your make sure to get your shot. We will never know when this pandemic will end. Wishing you and your family the best of health. Keep safe everyone. COVID-19 complication. What does COVID-19 vaccine efficacy mean? Since the onset of vaccine distribution, you have likely heard that the Moderna and Pfizer BioNTech COVID-19 vaccines have 94% and 95% efficacy rates. These percentages represent the vaccine's effectiveness. This video is brought to you by HealthyRN Ways. Videos will be posted every Monday, Wednesday and Friday about the whole COVID-19 pandemic. Please see my other videos at the end card of this video. That vaccine effectiveness refers to the proportionate reduction in cases among participants that have been vaccinated during a clinical trial. It's measured by calculating the risk of disease among vaccinated and unvaccinated people, and then determining how the disease reduction risk percentage compares among the two groups. The formula for this calculation is risk of contracting illness among unvaccinated, placebo, group minus the risk among vaccinated group, over the risk among unvaccinated groups. The greater the percentage reduction of illness in the vaccinated group, the greater the vaccine effectiveness. But what do these percentages mean for your safety from the virus? The currently authorized COVID-19 vaccine efficacy rates are high and comparable to other vaccines, like the chickenpox vaccine. Even COVID-19 vaccines with lower efficacy rates are effective at preventing severe illness and transmission of the virus. When any COVID-19 vaccine becomes available to you, you should get it if you can. While the vaccines are not 100% perfect at preventing COVID-19, the Pfizer and Moderna vaccines have high efficacies that are similar to other vaccines. For comparison, the 2 recommended doses of the varicella, chickenpox vaccine are 88 to 98% effective in providing immunity against any form of varicella, and 95 to 100% effective in preventing severe varicella and other vaccine available in the market worldwide. It is extremely difficult for a vaccine to have 100% effectiveness due to a myriad of factors, including whether the full dosing schedule was received, timing between doses, the ability of a person's immune system to develop antibodies, proper handling of the vaccine. Some vaccines, such as the AstraZeneca COVID-19 vaccine, not yet authorized for use in the US, it is available in other countries, only yields 62% efficacy. Despite this 30% difference, that vaccines, no matter their rate of effectiveness, slow the spread of the virus. If you receive the vaccine and did not develop an immune response, no real harm is done. If you contract the virus or spread it to someone who is unable to fight it off, the harm can be significant. It is best to err on the side of caution and obtain the vaccine. It's easy to get caught up in the effectiveness percentages. But these vaccines are incredibly effective at keeping people alive. All the percentages refer to disease protection. They are protective against severe disease and completely protective against hospitalizations and deaths. When the virus enters the person's body, they will have the antibodies that prevent the virus from situating itself in the body and preventing symptoms or an illness. Moderna reported that no cases of severe COVID-19 resulting in hospitalization or death were reported in the vaccinated group. The Pfizer-BioNTech vaccine reported 5 hospitalizations in the placebo group and 0 in the vaccine group. If a person gets the COVID-19 vaccine, but still ends up getting sick, the vaccine still counts as effective. If you contract COVID-19 after only a single dose of the vaccine or immediately after your second dose, this does not mean your vaccine was ineffective. The immune system takes time to develop antibodies after the vaccine, which can take up to 2 weeks after the second dose. If you get sick before your immune system has enough time to respond to the vaccine, this does not mean that the vaccine was ineffective. Everyone should get the vaccine in order to decrease overall levels of the virus. Just get vaccinated, because the more people that get vaccinated, the closer we'll get to herd immunity. What happens after vaccination? That despite getting the vaccine, people may still transmit it to others. Patients that get the vaccine may still be able to colonize. They may have the virus up in their nose and it may not cause them disease where they feel symptoms. Because the disease might still be transmitted even after vaccination, people continue wearing a mask, social distancing, and washing their hands regularly. If you have the opportunity to obtain your COVID-19 vaccine, ensure that you're able to receive both doses to allow for an adequate immune response to the vaccine. In between doses, and for up to 2 to 3 weeks after your second dose, we recommend you continue to take the recommended precautions as if you were still unvaccinated, since your immune system has not yet built an adequate immune response to protect against contracting the illness. Antibodies from vaccines versus antibodies from natural infection. Similarities and potential differences. Many people are interested in antibodies, proteins made by the immune system in response to an infection or vaccination. Antibody response is one of the key signs that a person was previously infected, or vaccinated, for a disease such as COVID-19. And sometimes, but not always, antibodies are a signal that an individual is protected from future infection. What are antibodies? Antibodies are proteins present on the surface of important cells of your immune system called B cells. B cells also release antibodies, partly with the help of another type of immune cell, T cells, and other system involved. Role in defeating initial infections. Antibodies play a key role in defeating certain types of infections. Through a variety of different mechanisms, in coordination with other parts of your immune system, some antibodies can inactivate and help eliminate pathogens. We think that includes the virus that causes COVID-19, SARS-CoV-2. It takes a while for this to work. If your immune system has never dealt with a particular virus before, it won't have antibodies to the virus ready to go. Antibodies bind very precisely to a specific spot on a given virus. It takes your immune system a while to figure out what exact antibody will work to neutralize a virus, or other type of pathogen. That's one of the reasons it takes you a while to get better after you are infected with a new virus. Depending on the specific type of antibody, it can take a couple of weeks or so to get the right antibodies produced in large amounts. A specific antibody type called Igm antibodies are usually the first to be produced. Detection of Igm antibodies can sometimes be used as a test for recent infection. For example, an Igm antibody to a specific protein is commonly used to check for recent infection with the hepatitis B virus. Other types of antibodies are usually produced a little later. A particularly important type is Igg antibodies, which tend to be more long-lived than Igm antibodies. This subtype of antibodies is critical not just for controlling initial disease but for preventing future disease if you are re-exposed in the future. Role in preventing future infections. After an infection, certain T cells and B cells that can recognize the virus stick around for a long time. If they are ever re-exposed to the virus, or other pathogen, again, these special memory cells recognize it quickly and start to respond. This helps the immune system be effective much more quickly. This way, you don't get sick. Or, if you do get sick, you usually only get a very mild version of an illness. When this happens, you are said to have protective immunity to a disease. Depending on the situation, immunity might last for months or years. You might also have partial immunity, which offers you some degree of protection, and a head start to the immune system if you get re-exposed and reinfected, but not total protection. Antibodies in COVID-19. It's because of this key role in both treating infection and preventing disease that scientists have been so interested in the role of antibodies in COVID-19. One of the treatments being given to some COVID-19 patients as a part of clinical trials is the plasma donated from people who have recovered from the illness. The idea is that that plasma contains antibodies to the virus that might help individuals recover more quickly from infection. Researchers are also hard at work developing cutting-edge synthetic antibody therapies that might end up being an important part of treatment. Antibody products have already received emergency use authorization by the FDA. These might be especially helpful early in the disease course. Studying how antibodies work in COVID-19 has also been critical for developing a successful vaccine. Knowledge about antibodies will be important for assessing how immunity to COVID-19, either from infection or from a vaccine, might decrease over time. From this we will learn when people might need booster vaccine shots to re-up their immunity. Even though antibodies are probably the most important means of inducing immunity in COVID-19, they may not be the only part of the immune system with an important role. For example, certain T cells play a role in protective immunity for some infections. This will become clearer with time. Neutralizing versus non-neutralizing antibodies. One confusing thing is that even though antibodies are important for eliminating and preventing many kinds of infections, not all antibodies the body produces against a virus are effective. Different B cells in the body will produce multiple different antibodies that bind to different sites on the body. But only binding to some of these sites will actually inactivate the virus. For a vaccine to work, it must produce this type of neutralizing antibodies. What about antibodies from natural infection? When you develop antibodies through natural infection, your immune system goes through this process of identifying the virus and eventually making effective antibodies. Your B cells make antibodies to different parts of the virus, some of which are effective and some of which are not. These help you eliminate the virus and recover. Hopefully, some of these antibodies also help protect you from future infection. Because the virus that causes COVID-19 is so new, there is still a lot that we don't know about it. But it seems like infection with COVID-19 gives you a relatively high degree of protection from being reinfected, at least in the short term. Very few cases of reinfection with SARS-CoV-2 have been documented worldwide. Because the virus is so widespread, if getting infected didn't give at least some protection, you'd expect many more people to have gotten the virus twice. Studies have indicated that people with symptoms of COVID-19 seem to produce antibodies, effective, neutralizing antibodies, as assessed in a lab. From our experience with other viruses, we think that that means that getting infected with COVID-19 probably leads to at least some level of protection against future infection. Additionally, studies in animals suggest at least some level of protective immunity, with at least some of this coming from antibody protection. How long might natural immunity last? How long that immunity lasts are a very important question. Different types of viruses differ in how long protective immunity lasts after infection. Some viruses mutate rather quickly. When you're exposed to a new strain of the virus, your previous antibodies might not work. Immunity to some types of coronavirus may be short-lived, as people can get cold-like symptoms from certain coronaviruses season. But coronaviruses don't mutate as rapidly as viruses like influenza, which cause the flu. This might mean that protective immunity might last longer for COVID-19 than it does for something like the flu. Antibodies to the new coronavirus do seem to decrease in the months after infection. That happens for all infectious diseases. It doesn't necessarily mean that immune protection is decreasing. The B cells actively releasing the relevant antibody may decrease their production in the months after an infection. But memory B cells can continue to circulate in the bloodstream for years in other types of infections. Presumably, these B cells could again start releasing the relevant antibody if they were again exposed to the virus. After they have studied a virus for a long time, scientists can establish certain standards for whether a person is immune based on laboratory standards that can be checked with a blood test, a certain concentration of a specific antibody. Because the virus is so new, we'll have to see what it looks like over time. 3 months after experiencing symptoms from COVID-19, one study found antibodies in the majority of people. Based on information from related viruses, some scientists estimate that immunity from natural infection might last 1 to 3 years. But the virus hasn't been around long enough for scientists to fully assess this. It might also make a difference whether one had an asymptomatic, mild, or a severe infection. What about antibodies from vaccination? Vaccination is a way for your body to build protective immunity without having to get sick first. Different types of vaccines do this in different ways. But in all cases, the immune system is exposed to one or more proteins from the virus, or other pathogen. That allows your immune system to make B cells that make specific antibodies that can neutralize that specific virus. The process of vaccination allows the formation of the memory B cells, just like they do in natural infection. If you're ever exposed to the virus, these B cells go into action right away and release antibodies that can target the virus. They inactivate the virus before you get sick. Or, in some cases, you might get sick but with a much milder case. That's because your immune system already has a head start, one it wouldn't have had if you hadn't been vaccinated. There are a lot of similarities but also sometimes some differences in the type of antibody and immune response you get from vaccination compared to a natural infection. As in response to a live virus, IgM-type antibodies usually come first, followed by IgG and some other types of antibodies. And just like in a natural infection, protective immunity doesn't begin the moment you get vaccinated. It takes a couple of weeks or so for your immune system to form the antibodies and groups of B cells that it needs. That's why you don't get full protective coverage from a vaccination right away. For the most part, the antibodies that you form from getting vaccinated are the same kind of antibodies you would get from a natural infection. One difference is that certain types of vaccines only show the immune system part of the relevant virus. Because of that, the immune system doesn't form as many different types of antibodies as it would in the course of a natural infection. Sometimes researchers can use this understanding to help make diagnostic decisions. Differences in certain antibodies can sometimes be used to determine if a person has an active or chronic infection with hepatitis B or if they have been successfully vaccinated. People who got the antibodies through natural infection have a specific antibody not found in people who got vaccinated, one not important for developing immunity. Most of the vaccines under development for COVID-19 only show the immune system part of the virus, a protein chosen to prime a strong immune response. This includes the Pfizer mRNA vaccine. Someone who had naturally been infected with the virus might have some additional antibody types not found in someone who had been successfully vaccinated. Assessing differences in natural versus vaccine-induced immunity. An important topic for researchers are these potential differences in the protective immune response, including antibodies, between people who got an infection naturally and people who got a vaccine. It is a very complex topic. You can't just compare natural infection to vaccination, because not every vaccine has the same properties, and not every vaccine will trigger exactly the same immune response. A specific vaccine might not provide as effective of an antibody response as being naturally infected. But other times, the reverse might be the case, especially if a vaccine has been especially designed to provoke a strong response. We can't make assumptions without studying the specific data over the long term. Potential risks of antibodies. We usually think about the benefits of antibodies in terms of eliminating infections and providing protective immunity. However, in rare circumstances, binding of an antibody might actually worsen an infection. Antibodies might bind to a virus in such a way that help it enter cells more easily. This might mean that a person reinfected after an initial mild infection might then have more severe disease. Theoretically might mean that a person could have a worse response to a potential infection with COVID-19 if they have previously been vaccinated for the disease. This scenario has been called antibody-dependent enhancement. It has been found in viruses such as dengue, in which it complicated the creation of successful vaccines. In some, but not all, animal studies, it has also been observed in a coronavirus closely related to the one that causes COVID-19, the virus that causes SARS. Because they were aware of this theoretical possibility, researchers have been looking very carefully to see whether this might be a possibility in COVID-19. No signs of antibody-dependent enhancement have been found in COVID-19. This includes the Pfizer mRNA vaccine for COVID-19, the only vaccine as of mid-December 2020 that has been released under an emergency use authorization by the FDA. Researchers will continue to monitor the effects of this vaccine and the others under development. With time, we'll get more data that will hopefully definitively quash this theoretical concern. We'll also continue to learn how immunity and the antibody response changes over time, both after natural infection and after vaccination with different types of COVID-19 vaccines. The available COVID-19 vaccines are highly effective, and so far, especially effective at preventing hospitalizations and deaths, specifically. A vaccine is still considered effective even if you get sick shortly after because the immune system takes time to develop antibodies. How Galilea's COVID-19 vaccine works. The Galileo Research Institute, part of Russia's Ministry of Health, developed a coronavirus vaccine known as Sputnik V or Igm COVID-Vac. Galileo published a study in February 2021 showing the vaccine had an efficacy rate of 91.6%. Russia is using it in a mass vaccination campaign, and it is now being distributed in Argentina, Belarus and other countries. This series of Race to Vaccine for COVID-19, brought to you by HealthyRN Ways. Videos will be posted every Monday, Wednesday and Friday about the whole COVID-19 pandemic from the start until the end of this pandemic. Please see my other videos about COVID-19 vaccines available now. Click the link at the end of this video. A piece of the coronavirus, the SARS-CoV-2 virus, is studied with proteins that it uses to enter human cells. These so-called spike proteins make a tempting target for potential vaccines and treatments. Sputnik V is based on the virus's genetic instructions for building the spike protein. But unlike the Pfizer BioNTech and Moderna vaccines, which store the instructions in single-stranded RNA, Sputnik V uses double-stranded DNA. DNA inside adenoviruses. The researchers developed their vaccine from adenoviruses, a kind of virus that causes colds. They added the gene for the coronavirus spike protein gene to two types of adenovirus, one called AD26 and one called AD5, and engineered them so they could invade cells but not replicate. Sputnik V comes out of decades of research on adenovirus-based vaccines. The first one was approved for general use last year, a vaccine for Ebola, made by Johnson & Johnson. Some other coronavirus vaccines are also based on adenoviruses, such as one from Johnson & Johnson using AD26, and one by the University of Oxford and AstraZeneca using a chimpanzee adenovirus. Entering a cell after Sputnik V is injected into a person's arm, the adenoviruses bump into cells and latch onto proteins on their surface. The cell engulfs the virus in a bubble and pulls it inside. Once inside, the adenovirus escapes from the bubble and travels to the nucleus, the chamber where the cell's DNA is stored. The adenovirus pushes its DNA into the nucleus. The adenovirus is engineered so it can't make copies of itself, but the gene for the coronavirus spike protein can be read by the cell and copied into a molecule called messenger RNA, or mRNA. Building spike proteins, the mRNA leaves the nucleus, and the cell's molecules read its sequence and begin assembling spike proteins. Some of the spike proteins produced by the cell form spikes that migrate to its surface and stick out their tips. The vaccinated cells also break up some of the proteins into fragments, which they present on their surface. These protruding spikes and spike protein fragments can then be recognized by the immune system. The adenovirus also provokes the immune system by switching on the cell's alarm systems. The cell sends out warning signals to activate immune cells nearby. By raising this alarm, Sputnik V causes the immune system to react more strongly to the spike proteins. Spotting the intruder when a vaccinated cell dies, the debris contains spike proteins and protein fragments that can then be taken up by a type of immune cell called an antigen-presenting cell. The cell presents fragments of the spike protein on its surface. When other cells called helper T cells detect these fragments, the helper T cells can raise the alarm and help marshal other immune cells to fight the infection. Making antibodies, other immune cells, called B cells, may bump into the coronavirus spikes on the surface of vaccinated cells, or free-floating spike protein fragments. A few of the B cells may be able to lock onto the spike proteins. If these B cells are then activated by helper T cells, they will start to proliferate and pour out antibodies that target the spike protein. Stopping the virus, the antibodies can latch onto coronavirus spikes, mark the virus for destruction and prevent infection by blocking the spikes from attaching to other cells. Killing infected cells, the antigen-presenting cells can also activate another type of immune cell called a killer T cell to seek out and destroy any coronavirus-infected cells that display the spike protein fragments on their surfaces. 2 doses. Some researchers worry that our immune systems could respond to an adenovirus vaccine by making antibodies against it, which would render a second dose ineffective. To avoid this, the Russian researchers used one type of adenovirus, Ad26, for the first dose, and another, Ad5, for the second. Adenovirus-based vaccines for COVID-19 are more rugged than the mRNA vaccines from Pfizer and Moderna. DNA is not as fragile as RNA, and the adenovirus's tough protein coat helps protect the genetic material inside. As a result, Sputnik V can be refrigerated and does not require very low storage temperatures. Remembering the virus, Gamaleya has announced that Sputnik V has an efficacy rate of 91.4%, but has not yet published a scientific paper with the full details of the trial. It's not yet clear how long the vaccine's protection might last. The level of antibodies and killer T cells triggered by the vaccine may drop in the months after vaccination. But the immune system also contains special cells called memory B cells and memory T cells that may retain information about the coronavirus for years or even decades. Clinical trials. Before the start of clinical trial, the vaccine had gone through all stages of preclinical trials with experiments on different types of animals, including 2 types of primates. Phase 1 and 2 clinical trials of the vaccine have been completed on August 1, 2020. All the volunteers are feeling well. No unforeseen or unwanted side effects were observed. The vaccine induced strong antibody and cellular immune response. Not a single participant of the current clinical trials got infected with COVID-19 after being administered with the vaccine. The high efficacy of the vaccine was confirmed by high-precision tests for antibodies in the blood serum of volunteers, including an analysis for antibodies that neutralize the coronavirus, as well as the ability of the immune cells of the volunteers to activate in response to the spike S protein of the coronavirus, which indicates the formation of both antibody and cellular immune vaccine response. Post-registration clinical trials involving more than 31,000 people in Russia and Belarus were launched on August 25, 2020. A number of countries, such as UAE, India and Venezuela, joined the clinical trials of Sputnik V locally. The vaccine has received a registration certificate from the Russian Ministry of Health on August 11 and under emergency rules adopted during the COVID-19 pandemic can be used to vaccinate the population in Russia. In Phase 3 clinical trials, Sputnik V showed strong efficacy, immunogenicity and safety results. Efficacy of Sputnik V against COVID-19 was reported at 91.6%. The figure is based on the analysis of data on 19,866 volunteers, who received both the first and second doses of the Sputnik V vaccine or placebo at the final control point of 78 confirmed COVID-19 cases. Sputnik V provides full protection against severe cases of COVID-19. Sputnik V guarantees robust humoral antibodies which are first line of defense, and cell-mediated, long-term protection, immune response. The Phase 3 clinical trial results proved high levels of safety and immunogenicity of the Sputnik V vaccines including elderly group. Over 98% of volunteers in the vaccine group developed humoral immune response and 100% cellular immune response. The level of virus-neutralizing antibodies of volunteers vaccinated with Sputnik V is 1.3x1. 5x times higher than the level of antibodies of patients who recovered from COVID-19. Vaccine efficacy for the elderly group was shown at 91.8% and did not differ statistically from the 18 to 60 group. The vaccine showed excellent safety profile. Most adverse events, 94%, were mild and included flu-like syndromes, injection site reactions, headache and asthenia. No serious adverse events associated with the vaccination, as confirmed by Independent Data Monitoring Committee. No strong allergies, no anaphylactic shock. The unique substance of the Sputnik V and method of using it has a patent protection in Russia, obtained by Gamaleya National Research Institute of Epidemiology and Microbiology. Vaccine timeline: June 2020 Gamaleya launches clinical trials of their vaccine, initially called GAM COVID-Vac. August 11 President Vladimir V Putin announces that a Russian healthcare regulator had approved the vaccine, renamed Sputnik V, before Phase 3 trials had even begun. Vaccine experts decry the move as risky. August 20 Russia walks back its earlier announcement, saying the vaccine approval was a conditional registration certificate that depends on positive results from Phase 3 trials. September 4 Gamaleya researchers publish the results of their Phase 1 half trial. In a small study, they found that Sputnik V yielded antibodies to the coronavirus and mild side effects. September 7 a Phase 3 trial begins in Russia. October 17 a Phase 2/3 trial launches in India. November 11 the Russian Direct Investment Fund announces the first preliminary evidence from their Phase 3 trial indicating that the vaccine is effective. Based on 20 cases of COVID-19 among the trial participants, Russian scientists estimate that the vaccine has 92% efficacy. November the Russian government begins offering Sputnik V within Russia in a mass vaccination campaign. But worry that the vaccine was rushed to approval leads to widespread hesitancy in the country. December the Phase 3 trial reaches its final total of 78 cases. The efficacy rate was effectively unchanged, at 91.4%. Out of the 78 cases of COVID-19 in the trial, 20 were severe, and all 20 were in volunteers who received the placebo. In addition, the researchers announced that they found no serious side effects from the vaccine. December 11 the drug maker AstraZeneca, which is developing an adenovirus-based vaccine in partnership with the University of Oxford, joins forces with Gamaleya to see if combining their vaccine with Sputnik V would increase the efficacy of the Oxford-AstraZeneca vaccine. December 24 the Associated Press reports that trial volunteers who suspect they received the placebo are dropping out to receive the vaccine now that it's widely available. The researchers running the trial reduce its planned size from 40,000 to 31,000 participants, causing experts to worry that it will not have enough statistical power to reach strong conclusions about the safety and efficacy of the vaccine. December 22 Belarus becomes the first country outside of Russia to register Sputnik V. December 23 Argentina authorizes the vaccine for emergency use. December 24, AstraZeneca registers a Phase 1 trial for a combination of the Sputnik V and Oxford AstraZeneca vaccines. To end the acute phase of this pandemic, and to control the virus in the longer term, the world needs multiple safe and effective vaccines which can be deployed in a range of populations and countries. This race to COVID-19 vaccine is in full swing. Please watch my videos on other COVID-19 vaccines manufacturers. How Clover Biopharmaceuticals and Dynavax COVID-19 vaccine is made. Adjuvanted S-trimer COVID-19 vaccine candidates demonstrated favorable safety and tolerability profiles and strong neutralizing immune responses in a Phase 1 trial. Clover plans to initiate a global Phase 2/3 trial in the first half of 2021 with an interim analysis for vaccine efficacy potentially in the middle of 2021. This series of Race to Vaccine for COVID-19, brought to you by HealthyRN Ways. Videos will be posted every Monday, Wednesday and Friday about the whole COVID-19 pandemic from the start till the end. Please see my other videos about COVID-19 vaccine, click the link at the end of this video. Clover Biopharmaceuticals, Clover, a global clinical stage biotechnology company committed to developing transformative biologics as vaccines and therapeutics for the world's most debilitating diseases, and Dynavax Technologies Corporation, Dynavax, Nasdaq, DVAX, a biopharmaceutical company focused on developing and commercializing vaccines, today announced the plan to initiate a global Phase 2/3 efficacy trial with the S-trimer COVID-19 vaccine candidate adjuvanted with CpG 1018 plus alum in the first half of 2021 with an interim analysis for vaccine efficacy potentially in the middle of 2021. The Coalition for Epidemic Preparedness Innovations, CEPI, will continue to support the development of Clover's COVID-19 vaccine candidate and will fund the development, including the Phase 2/3 trial, through licensure. In its Phase 1 clinical trial, Clover's protein-based COVID-19 S-trimer vaccine candidates in combination with adjuvants from either Dynavax or GlaxoSmithKline plc, GSK, London Stock Exchange, GSK, both performed well and induced high levels of neutralizing antibodies while demonstrating favorable safety and tolerability profiles. Clover expects to produce hundreds of millions of vaccine doses in 2021 and up to 1 billion vaccine doses in peak annual production is required by global demand. Given the encouraging results of our adjuvanted S-trimer COVID-19 vaccine candidate to date, we are enthusiastic about progressing to a global Phase 2/3 efficacy study utilizing Dynavax's advanced adjuvant CpG 1018 plus aluminium. We believe this vaccine candidate could be efficacious while potentially having a differentiated, beneficial reactogenicity and safety profile which could make it attractive for a broad population of peoples. We will continue to work closely with our collaborators and regulatory authorities worldwide to help make our vaccine accessible to those most in need as quickly as possible. We are excited to be rapidly progressing to an efficacy trial to support filing for emergency use and approval. Clover's S-trimer antigen adjuvanted with CpG 1018 plus alum demonstrated low reactogenicity while providing high levels of neutralizing antibodies and a strong Th1 biased cell-mediated immune response. We are proud to be collaborating with Clover on the development of this vaccine for COVID-19 and committed to supporting Clover in making the vaccine available globally. Clover and GSK also decided to discontinue their partnership to evaluate the S-trimer COVID-19 vaccine candidate with GSK's pandemic adjuvant system. The companies all remain united in their missions to maximize their contributions to ending the current pandemic. All parties remain confident that their respective strategies and collaborations for COVID-19 vaccine development will be able to maximize their respective and overall impacts against the COVID-19 pandemic. About S-trimer COVID-19 vaccine, utilizing Clover's proprietary trimer tag technology, S-trimer is a trimeric SARS-CoV-2 spike, S, protein subunit vaccine candidate. Similar to other enveloped RNA viruses such as HIV, RSV and influenza, SARS-CoV-2 is also an RNA virus that has a trimeric spike, S protein on its viral envelope. The trimeric S protein of SARS-CoV-2 is responsible for binding to host cell surface receptor ACE2 and subsequent viral entry, making it the primary target antigen for vaccine development. S-trimer resembles the native trimeric viral spike protein and is produced via a rapid mammalian cell culture-based expression system. S-trimer is intended to be adjuvanted. About Trimer Tag technology is an innovative drug development platform which allows the production of novel, covalently trimerized fusion proteins. Many major disease targets are trimerization dependent, such as the tumor necrosis factor, TNF, superfamily, involved in extrinsic apoptosis, immune co-stimulation and inflammation, as well as enveloped RNA virus antigens responsible for entry into host cells. Clover is using its Trimer Tag technology with global IP position to develop recombinant trimerized fusion proteins that are able to effectively target these previously undruggable pathways. About Clover Biopharmaceuticals is a global, clinical stage, research-based biotechnology company focused on discovering, developing and commercializing transformative biologic therapies, with a focus on oncology and autoimmune diseases, as well as viral vaccines. Having raised more than $350 million in total capital since 2016, Clover is utilizing its proprietary Trimer Tag technology platform to develop novel biologics targeting trimerization-dependent pathways. Additionally, Clover is leveraging its in-house GMP biomanufacturing capabilities, which is certificated by Qualified Person (QP) within the European Union (EU), to support large-scale production of its biologic therapies. About CpG 1018 is the adjuvant used in HEPLISAVB, hepatitis B vaccine, recombinant, adjuvanted, and adult hepatitis B vaccine approved by the U.S. Food and Drug Administration, FDA. Dynavax developed CpG 1018 to provide an increased vaccine immune response, which has been demonstrated in HEPLISAVB. CpG 1018 provides a well-developed technology and a significant safety database to support the rapid development and large-scale manufacturing of a COVID-19 vaccine. Upon completion of ongoing scale-up activities, the existing equipment capacity for CpG 1018 will be 600 million to 1.2 billion adjuvant doses annually, depending on final dose selected. About Dynavax is a commercial-stage biopharmaceutical company developing and commercializing novel vaccines. The company's first commercial product, HEPLISAVB, hepatitis B vaccine, recombinant, adjuvanted, is approved in the US for prevention of infection caused by all known subtypes of hepatitis B virus in adults age 18 years and older. Dynavax is also advancing CpG 1018 as a premier vaccine adjuvant through research collaborations and partnerships. Current collaborations are focused on adjuvanted vaccines for COVID-19, pertussis and universal influenza. About CEPI is an innovative partnership between public, private, philanthropic, and civil organizations, launched at Davos in 2017, to develop vaccines to stop future epidemics. CEPI has moved with great urgency and in coordination with WHO in response to the emergence of COVID-19. CEPI has initiated 11 partnerships to develop vaccines against the novel coronavirus. The programs are leveraging rapid response platforms already supported by CEPI as well as new partnerships. Before the emergence of COVID-19, CEPI's priority diseases included Ebola virus, Lassa virus, Middle East respiratory syndrome coronavirus, Nipah virus, Rift Valley fever and chikungunya virus. CEPI also invested in platform technologies that can be used for rapid vaccine and immunoprophylactic development against unknown pathogens, Disease X. Dynavax forward-looking statements. This press release regarding the potential to develop a COVID-19 vaccine containing CpG 1018. Actual results may differ materially from those set forth in this press release due to the risks and uncertainties inherent in vaccine research and development, including the timing of initiating clinical trials and completing development, whether CpG 1018 plus aluminium combined with Clover's protein subunit vaccine will prove to be beneficial in clinical trials, whether and when the vaccine will be approved for use, whether CEPI will continue to fund the Clover program through development and licensure, and whether sufficient quantities of CpG 1018 will be able to be manufactured, as well as other risks detailed in the Risk Factors section of our annual report on Form 10-K for the fiscal year ended December 31, 2020, as well as discussions of potential risks, uncertainties and other important factors in our other filings with the U.S. Securities and Exchange Commission. We undertake no obligation to revise or update information herein to reflect events or circumstances in the future, even if new information becomes available. We believe this vaccine candidate could be efficacious while potentially having a differentiated, beneficial reactogenicity and safety profile which could make it attractive for a broad population of peoples. We will continue to work closely with our collaborators and regulatory authorities worldwide to help make our vaccine accessible to those most in need as quickly as possible. The trimeric S protein of SARS-CoV-2 is responsible for binding to host cell surface receptor ACE2 and subsequent viral entry, making it the primary target antigen for vaccine development. S-trimer resembles the native trimeric viral spike protein and is produced via a rapid mammalian cell culture-based expression system. S-trimer is intended to be adjuvanted. Clover's vaccine candidate has the potential to be manufactured at scale and stored in a regular refrigerator, which makes it suitable for use around the globe, including in low-resource settings. Through this partnership, we hope to make hundreds of millions of doses of this vaccine globally accessible through COVAX if it is proven to be safe and effective. China-based Clover Biopharmaceuticals is a global, clinical-stage, research-based biotechnology company focused on discovering, developing, and commercializing transformative biologic therapies, focusing on oncology and autoimmune diseases, as well as viral vaccines. To end the acute phase of this pandemic, and to control the virus in the longer term, the world needs multiple safe and effective vaccines which can be deployed in a range of populations and countries. Clover's vaccine candidate has the potential to be manufactured at scale and stored in a regular refrigerator which makes it suitable for use around the globe, including in low-resource settings. Through this partnership we hope to make hundreds of millions of doses of this vaccine globally accessible through COVAX, if it is proven to be safe and effective. The race for COVID-19 vaccine is in full swing, many pharmaceutical companies doing non-stop researching and trials in developing vaccine all over the world. Please watch my videos, like, share and subscribe to my channels for more videos on coronavirus. Thank you for watching, see you on my next video. Ma Salama.